Good evening. How are you doing? Today I will talk about the difference between gram-positive and gram-negative bacteria. Look at this picture. This is a typical bacteria. A bacteria is a prokaryotic cell, which is an ancient bacteria. The term prokaryotic cell originates from prokaryote, which is before nucleus. Well, the bacteria was founded 3 billion years ago. The bacteria is made up of three main structures, which are, first of all, the plasma membrane, which is present, the inner membrane here in green. It's a selective membrane that controls everything that enters and exits the cell. Its nature is a phospholipid, which is made up of a type of lipids, which is very common in many membranes. Next, the most important structure is cell wall. The cell wall is a very rigid structure that provides support for the bacteria. It's made up of a peptidoglycan, which is a type of polysaccharide, which is made up of repetitive disaccharide sugars. You will see its structure in a moment. Third structure is the capsule. It's a sticky outer layer made up for protection and attachment. Here is a table that will summarize differences and the similarities between gram-positive and the gram-negative bacteria. Let's begin with the inner membrane. This is both gram-positive and the gram-negative has an inner membrane. This is how the phospholipid bilayer looks like, with hydrophilic heads and hydrophobic tails. So it's selective. It doesn't allow any foreign molecules to cross through it. Next, the peptidoglycan. It's a repetitive disaccharide sugar, as you can see. There is a thick layer here made up of like 5 layers in gram-positive bacteria, while in gram-negative bacteria, it's in only 2 layers. Next, we can see an additional outer membrane in gram-negative bacteria, not present in gram-positive bacteria. This is an important difference. Both gram-positive and gram-negative bacteria contains capsule. For gram-positive bacteria, it's more easily treatable with antibiotics. Why? Because there is no outer membrane, so the antibiotics can enter easily and fit into the inner membrane. While in gram-negative, the presence of the outer membrane, phospholipid lipid bilayer membrane, makes it harsh for the antibiotic to fit and enter into the inside membrane. Okay. Stain purple violet after gram staining. So in gram-positive bacteria, the color of the staining is purple violet, while in gram-negative, the color is reddish pink. We will see why is that and what is gram staining. Well, gram staining is a way to identify type of infectious bacteria. Treatment. It differs based upon outcome. First of all, bring an inoculation. This instrument is called inoculation loop. Bring it and insert it inside any medium contains bacteria and start moving it until some bacteria is attached to this inoculation loop. Then move this inoculation loop and place it inside a glass slide and put a smear of bacteria across this glass slide. Then, as you can see here, then what should you do? Heat fix the bacteria to the slide. You have to fix this bacteria to the slide by heating. As you can see here, we move the slide up right and left in order to make heat enter in order to heat it. Why should we heat it? In order to the bacteria not to get out while we wash it. Simply in the latter step, we're going to add some certain chemicals and washing, and we'll do washing, so the bacteria won't leave the slide. It will be stuck. It will stuck. Okay. Number three, apply crystal violet to the bacteria. So we add some drops of crystal violet to the bacteria. Let us zoom to the cells of the bacteria after adding the crystal violet. Well, after we here is just an example. This is gram positive and this is gram negative. We will understand why gram positive has crystal violet staining, why gram negative has a pink staining. Okay. Well, you can see that both gram positive and gram negative here contain crystal violets. Okay, so the crystal violet is in the inner membrane in both bacteria. Then we apply iodine to the bacteria. We will at the end discover which is gram positive and which is gram negative. Okay, so as you can see that the gram positive that the iodine and the crystal violet clumped together and formed like a larger molecule, as you can see. Now we are going to try to add to wash with alcohol. After washing with alcohol, we will analyze that in gram-positive bacteria, the cell wall did shrink, and obviously the capsule disappeared. What about gram-negative? The cell wall also disappeared, also shrinked, but the outer membrane disappeared and the capsule was also washed. This is because the alcohol has a dehydrating agent. Okay, so uh, so after we did that, we apply safranin to the bacteria. The safranin is a red pink, has a red pink color. So as you can see, it's stuck in the lipid bilayer, mainly the heads of the lipids. So at the end, the result is program. There's a purple appearance. Why is that? Well, because actually the pink color is masked by the purple. So the purple has a dominance color. That's why it appeared in gram positive, while in gram negative, there is actually no presence for crystal violet. Why? Because it's washed from the medium. Okay, it's washed because the thin layer of peptidoglycan didn't protect it. Yeah, so it's washed from the medium. So at the end, the gram-negative bacteria become pink. Okay, so finally, look at this. This is a picture of a gram-positive bacteria, which has a thick layer of purple. It has a purple color, while a gram-negative has a reddish pink color. Okay. So finally, this is a recap. So the proper treatment can begin once the results of gram stain are known. So when I discover that this is a gram-positive bacteria, the antibiotic is definitely more easily entering. The gram-positive will enter more easily to the gram-positive bacteria. Unlike the gram-negative bacteria, since it has a dual membrane, so it's definitely hard to enter a bacteria. Thank you so much for attending. Goodbye.